Assalamu alaikum. So today we are going to discuss the metabolism of carbohydrate. So in metabolism of carbohydrate, we will discuss glycolysis first, then link reaction. After that, PCA cycle. Then the product of all these three reactions is used up in oxidative phosphorylation. We're gonna discuss them step by step, one by one. So we say the ultimate source of energy is light. So during the photosynthesis, light energy is stored in the form of potential chemical energy in carbohydrate, glucose, in carbohydrates, especially in the form of glucose, lipids, and protein molecule. We say these three major organic compounds, carbohydrate, lipids, and protein, they contain the potential chemical energy. So this potential chemical energy is supposed to be released from these compounds and used up in a different, like say, different activities inside the body, like active transport, cell division, protein synthesis, where the energy is required. But this energy cannot be used up directly, so it's supposed to be converted to the ATP. So energy is converted to the ATP, then it can be used up for the muscle contraction, cell division, active transport, and various energy requirement processes inside the cells. So we say, first of all, we start with the glycolysis. Glucose molecule contain the potential chemical energy. That potential chemical energy is released. So we say in the first, like say, release in the three major steps of glycolysis. First of all, first step is known as phosphorylation of glucose. Second step is known as splitting of hexose into one bisphosphate molecule. And third step is oxidation of triose phosphate. So these are the three steps which are used for the like say which are used to break down the glucose molecule and ultimately into the pyruvate molecule. We would discuss them one by one. So we start with the first of all. Now we say these are the three major steps which are used in glycolysis. First step is known as phosphorylation of glucose. During the phosphorylation of glucose is attached with two phosphate molecules. So glucose molecule is converted to the hexose 1,6 bisphosphate. So first of all, we should know that glucose molecule enter into the cell. So this one is a glucose molecule. It will enter into the cell. Inside the cytoplasm of the cell, glycolysis would take place. So for the entry of the glucose into the cell, a special transport protein is required. These are known as glucose transport protein. We also use the word GLUT for this. Through the glucose transport protein, glucose would enter into the cytoplasm of the cell. Inside the cytoplasm, glycolysis would take place. So these three are the major steps of glycolysis. So once the phosphorylation of glucose, like say after the phosphorylation of glucose molecule become polar, it cannot come out of the cell. So it stays inside the cell. So it is used up in the process of glycolysis. We start with the process of glycolysis. During like the first step of the glycolysis is phosphorylation of glucose molecule. In the first step of phosphorylation of glucose molecule, glucose attached with one phosphate to give us glucose, like say glucose 6 phosphate. Glucose 6 phosphate is further isomerized to convert into fructose 6 phosphate. So we say this one is attachment, like say attachment of one more phosphate will take place. So will give us fructose 1,6 bisphosphate. So first of all, phosphorylation of glucose will take place. As a result of this phosphorylation of glucose attaches with two ATP molecules. So right over here, instead of gaining the energy, instead of making the energy, so two ATP molecule would be used up to give us hexose 1,6 bisphosphate. Hexose 1,6 bisphosphate is a 6-carbon sugar. It is attached with two phosphate molecule. Right? So right over here, glucose used up two ATP molecule. So give us hexose 1,6 bisphosphate, means 6 carbons attached with two phosphate. One is attached at carbon number 1, other one is attached to carbon number 6. So we say first step is phosphorylation of glucose, and the next step would be splitting of hexose 1,6 bisphosphate. In the first step, hexose 1,6 bisphosphate was formed. Now this hexose 1,6 bisphosphate would split into the two molecule of triose phosphate. How many molecule? Two molecule of triose phosphate would form. Now these two molecule of triose phosphate known as glycerol di3 phosphate, other one is known as dihydroxyacetone phosphate. So these glycerol di3 phosphate and dihydroxyacetone phosphate, they are interconvertible. Like say one can be converted to another one, but the use it for the use in the next step, glycerol di3 phosphate will be used up. So we say first step was the phosphorylation of glucose. And the second step is, like say, as a result of phosphorylation, hexose 1,6 bisphosphate is formed. Now this hexose 1,6 bisphosphate would split into the two molecule of triose phosphate. Triose phosphate means right over here, each carbon compound was, let's say each compound was containing 6 carbon. So right over here it contains, each molecule is contained three carbon atom and one phosphate. So we use the word triose phosphate. Right now this triose phosphate is formed. So there, like say in the third step of glycolysis, there will be oxidation of triose phosphate, mean oxidation of glycerol-3 phosphate. So during the oxidation of glycerol-3 phosphate, so coenzyme NAD is required, dehydrogenase enzyme is required. So oxidation of triose phosphate, it would give us, it was during the oxidation of triose phosphate, so it would give us two pyruvate molecule, or let's say as well as it would give us two ATP molecule and two NADH molecule. So we gonna give, like say, study them one by one. So right over we can say, during the oxidation of triose phosphate, as a result of oxidation of triose phosphate, so will give us one, like say we will give us, like during oxidation will give us one NADH, like say two NADH are formed because this reaction would take place twice. And like say during the each reaction two ATPs are formed, so ultimately will give us, like say, two ATPs are formed because there will be phosphorylation of two ADP during the each reaction. So we say, now we're going to link them one by one. So first of all, so these were the steps of glycolysis. If we consider the stages of glycolysis, during the stages of glycolysis, the first stage is phosphorylation of glucose, which would take place in the first step. So if you have to, like say, if you can have two scenarios in the examination as well. You can have the question stages of glycolysis, or you can have write down the steps of glycolysis. When the examiners say write down the step of glycolysis, you are supposed to give us these three steps. First of all is oxidation. First of all is phosphorylation. Second one is splitting of hexose 1,6 bisphosphate, and third step would be oxidation of triose phosphate. But if you have the question, write down the stages of glycolysis. During the stages of glycolysis, you are supposed to write down these 6 stages one by one. So first stage is, so these stages and steps, they are interlinked with each other. We can say steps are explained in the stages of glycolysis. We say phosphorylation of glucose would take place. So right over here, during the phosphorylation of glucose, one ATP would be used up right over here. Right, so glucose would convert into glucose 6-phosphate. After the conversion of glucose 6-phosphate right over here, now this glucose 6-phosphate, after the conversion of phosphate to the glucose, so this glucose molecule become activated, it can be used up in the glycolysis. So right over here, glucose 6-phosphate would convert into fructose 6-phosphate. So glucose and fructose, they are isomers of each other. So the next step would be isomerization of glucose 6-phosphate to fructose 6-phosphate. Now we say this fructose 6-phosphate will be further phosphorylated by using one more ATP. So we say fructose 6-phosphate would convert into fructose 1,6 bisphosphate. Fructose is a 6-carbon compound. So one, like 6-carbon compound, one phosphate is attached at carbon number 1, other one is attached at carbon number 6. So we use the word fructose 1,6 bisphosphate. So far we used up 2 ATP instead of making the 2 ATP. Now the next step would be cleavage of fructose 1,6 bisphosphate. This fructose 1,6 bisphosphate is a 6-carbon compound. It would, like say, it is broken down into two molecules. One of them is known as glycerol-3 phosphate and dihydroxyacetone phosphate. So these are known as trioses. So we say during triosis, the third step of within glycolysis, during triose, let's say these trioses, they are oxidized. Oxidation of trioses will take place. So during oxidation, glycerol-3 phosphate is converted into 1,3-bisphosphoglycerate. So right over here, during the conversion of glycerol-3 phosphate to 1,3-bisphosphoglycerate, one inorganic phosphate would attach with one inorganic phosphate would attach with glycerol-3 phosphate. So right over here, ATP is not used. So this phosphate is known as inorganic phosphate, which is already present inside the cytoplasm. It attaches, so will give us 1,3-bisphosphoglycerate. Right. So this one is known as PI, inorganic phosphate, which is already present inside the cytoplasm. So it attaches with glyceraldehyde 3-phosphate to give us 1,3-bis-phosphoglycerate, mean this triose would consist of 3 phosphate molecules. So right over here, when the one with phosphate, so same time, one phosphate is attached, so same time two hydrogen molecules are removed, so will give us NADH2. So we, right over here in the step 5, 1 and 2, let's say one molecule of NADH2 is produced. So and then as well as 1,3-bisphosphoglycerate is produced. So right over here, 3 carbon compounds attached with 2 phosphate. So one of the phosphate is removed. So this removal of phosphate, so the phosphate is which was removed, which is going to be removed from this, like say glycerol, this phosphoglycerate is taken up by the ATP. Now we can say we have right over here, we can say in the step 5, we were having the compound glyceraldehyde 3-phosphate. Which compound? Glyceraldehyde 3-phosphate. This glyceraldehyde 3-phosphate dehydrogenase and coenzyme right over here. So they would convert. So we can say stages of glycolysis comprise of these 6 steps. So we're gonna discuss them. These 6 stages of glycolysis one by one. So first stage is phosphorylation of glucose. So 1 ATP is used up over here during phosphorylation of glucose. So this glucose is converted to glucose 6-phosphate. After the conversion of glucose 6-phosphate, it is activated now. So we can say now it can be used up in the further process of glycolysis. The next step would be isomerization of glucose 6-phosphate to fructose 1 to fructose 6-phosphate. Mean glucose and fructose are isomers of each other. Definition of isomer is so they have the same molecular formula but different structure formula. The next step would be phosphorylation of fructose 1 to fructose 6-phosphate. So as a result of phosphorylation of fructose 6-phosphate, one more phosphate is attached with the fructose to give us fructose 1,6-bisphosphate. So right over here, 2 ATP are used up. So one ATP was used in the beginning of phosphorylation of glucose molecule. The second ATP would be used up for the phosphorylation of fructose 6-phosphate to give us fructose 1,6-bisphosphate. Now this fructose 1,6-bisphosphate is 6 carbon compound. It would split up into the three molecule of trioses. So fructose, cleavage of fructose 1,6-bisphosphate would take place. So as a result of this, glyceraldehyde 3-phosphate is produced. Another molecule is known as dihydroxyacetone phosphate. So now these two glyceraldehyde 3-phosphate, dihydroxyacetone phosphate, they are interconvertible. They are interconvertible molecule. So now over here in the stage 5, glyceraldehyde 3-phosphate would convert into 1,3-bisphosphoglycerate. So glyceraldehyde 3-phosphate is a the 3 carbon compound. So it is again converted. One more phosphorylation would take place. One more phosphate is attached. But right over here, this phosphate is inorganic phosphate. So right over here in this attachment, there is no utilization of ATP. Inorganic phosphate attached with glyceraldehyde 3-phosphate. So give us 1,3-bisphosphoglycerate. So as well as over here, we also give us one reduced NAD is produced as well. So NAD will convert into NADH2. So as a result of this, reduced NAD is produced, and one one inorganic phosphate is used up. So give us 1,3-bisphosphoglycerate. Now this 3 carbon compound would contain 2 phosphate molecules. So as a result of this, when 1,3-bisphosphoglycerate molecule would convert into 3-phosphoglycerate. So one phosphate is detached from the 1,3-bisphosphoglycerate. That phosphate is taken up by the ADP, adenosine diphosphate, to give us ATP. So right over here, one ATP is formed, right? So in the next molecule, so this reaction is, by say this reaction is cycled twice. So give us 2 ATP. Again over here now, this 3-phosphoglycerate would convert into 2-phosphoglycerate. This 2-phosphoglycerate would convert into pyruvate. So right over here again, one ADP is converted into ATP. So this during the glycolysis of one glucose molecule, this process would be revised twice.